Thank you, Brandon, and it's lovely to be here. As Brandon says, I'm going to talk about nature and data and beauty in those. And for me, as also for colleagues that I work with, nature and data come together under the topic of biophysics. And what I'm going to say, happily, is going to follow on nicely from the previous three presenters. So, and in terms of what I want to talk about, I'm actually going to group the points I want to make, which, as I say, come very much from my personal point of view, under a series of more general terms, which you will find used in all sorts of other kinds of discourse, because I think they are helpful ways to think about the kinds of applications of perceptions of beauty that are going on in these different areas of science. And the first thing I'm going to talk about is a couple of examples of idealism. This also happily follows on from Mylena, because this actually relates to the Mieselson-Stahl experiment that she was talking about. So on the left-hand side of the screen, you see a famous cartoon drawn by Watson and Crick for their paper published in 1953 describing the structure of DNA. And if you read that paper, a very short paper, which you'll be able to find on the Nature website, I'm sure perhaps you've all read it, you will remember if you've read it, or you'll find if you go and read it, that it's framed very much in ideal terms. It's very much about what would be neat given the data we've got. There were certain bits of information they had, but they needed to put them together into a satisfying whole, and they came up with the double helix hypothesis, which turned out to be entirely correct, essentially, and which leads on, of course, on the right-hand side to the experimentally determined structures for DNA itself, and of the more complicated RNA, which has because of a particular chemical modification in ribose compared to deoxyribose, has a much more complex range of folding patterns. But at the core idea that Watson and Crick came up with was correct, that base pairing leads to the double helix. The reason it relates to Mylena's talk and to the Mieselson and Stahl experiment is that, again, very famously at the end of the 1953 paper, and sort of with their tongue in their cheek, Watson and Crick said, "It has not escaped our notice that this structure suggests a possible mechanism for the replication of DNA." And of course, their mechanism was exactly the mechanism which Mieselson and Stahl went on to show was correct, the semi-conservative hypothesis for DNA replication. So that was an idealistic process. Idealism 2 here is another example of, which is, in this case, relates to symmetry, I guess, as does the DNA example. Also, Watson and Crick, in 1956, published again in Nature a paper describing what they thought the structures of small viruses would be like, and they needed to solve the problem of how it is that you can enclose a genome with a protein that it's encoded, and this is a basic problem to do with the size of the molecules. And they came up with some simple ideas about how you could essentially organize triangles in order to generate closed structures which would enclose the genomes of viruses. They did it perhaps informed by knowledge about the platonic solids, which you can see some ancient dice down below replicating those, the 5 platonic solids, of which the one which gets the most complex structure with the simplest shape is the icosahedron, and that was indeed what they proposed would be the structure of small viruses. And this is essentially as a theoretical paper by Watson and Crick, which turns out when you solve the structure of viruses like the blue-tongue virus or any number of other isometric viruses, and this was a paper published, as you can see, 25 years ago or so by some colleagues of mine here in Oxford. The blue-tongue virus, 100 million Daltons, the largest structure to be solved at the time, 720 copies of one protein forming outer shell, 120 copies of another protein forming an inner shell, the DNA, the double-stranded RNA of the genome also being resolved. It essentially has this icosahedral symmetry, and so their theory is correct. But of course, all symmetry is not perfectly isometric, and so you can also play with the same ideas to come up with something which is more distorted, but nevertheless has the same core idealistic senses behind it. And in fact, the structure of the HIV capsid turns out to use that idea. It also, like icosahedra of an isometric form, has 12 pentagonal vertices, but they're arranged in a different set of positions to where you would find those 12 pentagonal vertices in blue-tongue virus, where they're arranged isometrically. And so in HIV, you get a distorted cone. And so you can see idealism 2 here is how people are playing with symmetry and ideas of what would be fitting, and we'll come on to fit in a more general sense in a moment, to come up with theories which turn out to be correct for how it is that nature is ordering itself chemically. And I want to go on to observation, and this relates directly to what Mylena was talking about. I want to say, first of all, something about acoustics on the left-hand side. So acoustics is here because I think that many experiments, if not all experiments, have a close relationship to play. And play, from my point of view, is an aesthetic experience. It's something that we enjoy and have delight in. And the reason why I choose acoustics is because many of the kinds of experiments that I would do or colleagues of mine would do involve taking our sample we want to study, placing it in some kind of medium, and exerting some kind of force on it, such as, for example, Marlene referred to centrifugation used in the measles and the Stahl experiment. What they did was that they centrifuged the DNA to separate it differentially depending on how dense it was, and that is placing their DNA in water and gravitationally spinning it, and then you get movement of the thing, and you follow how it moves, and that tells you something about the structure of the DNA. Acoustics is relevant because many of the systems that we study involve taking our system, placing it in a medium of different kind, and applying force to it. And that is very playful, actually, and it feels very playful. There's an enjoyment in grappling with the experiment, and so that's why acoustics is a good analogy. The diamond light source down the road at the Harwell Research Campus, Marlene also referred to the much larger experimental machinery we now use. The diamond light source is an example of one such system. It's a very bright source of tunable X-rays. It's how you determine structures like the virus structure I showed in the previous slide. And again, there is delight in the design of huge machines with highly sophisticated physics behind them, which enable you to achieve the biological question, answering the biological questions you wish to. But we can observe more directly, and you can see a movie down here taken using a technique called atomic force microscopy of a conformational change occurring in a protein that we work on as it in this case attacks bacteria, and you can see that there's a clockwise movement as it undergoes its conformational change, which is a brightening of the whole structure. So there's delight in observation as well, which I think feeds into our aesthetic experience in science. Another way in which aesthetics comes into the kind of data and nature area that I want to talk about, and particularly in biophysics, is that we can represent our systems in ways which people commonly will find aesthetically pleasing. We can depict structures of proteins like this perforin protein forming a pore. We can depict them as attractive ribbons. We can color them nicely. In a trivial sense, this is aesthetic, but people do kind of like it. They do warm to the representations that you can give. They will even sometimes ask you, are they really that color? And of course, they're not really that color. It's a way that enables us to try and depict an idea that we want to come across with. But this is pulling together a whole load of data we've got into a movie, which enables us to explain ourselves. Representation is another form of aesthetic process, in this case, express it, aesthetic kind of discourse, if you like, in biophysics. And then form kind of pervades the issue of how we see beauty in nature and data. The form of an alpha helix on the left-hand side there, the form of in this case, the SARS-CoV-2 spike, which is the bit of the protein that attacks our ACE2 proteins on human cells in order to insert into them. The forms which we might be able to relate to other forms in the world. So colleagues of mine studying the SARS-CoV-2 spike noticed that the receptor binding domain bore a resemblance to a torso, such as you might find in a Greek statue. And so that enabled them to organize their thinking as to where antibodies were binding and where the more powerful antibodies were binding. So you can see there's a kind of pooling together of different aesthetic senses in order to bring out some general ideas or some take-home messages that people wish others to understand from their work. And it's about the form of the information and how we can display it. And then fit. I said I would mention fit, and this is kind of relates also to things like DNA, where clearly we're talking about the fitting together of two strands. But fit, and this for me was how I really got into a sense of the beauty in science, and really what led me to become a biochemist. The fit of molecules together. This molecule, something called EDTA, bound to a divalent cation, a metal ion, or the fit of drugs to target enzymes we might be trying to inhibit, and that's a structure of Paxlovid, which is a commercially marketed, it's now available in the clinic, inhibitor of the SARS-CoV-2 polymerase, and that is now out there marketed. It's enabling people to better fight off the coronavirus. But what enabled that work to be done was solving structures and then aesthetically engaging with them and building models into those structures in order to understand how we could inhibit the enzyme, and having inhibited, then how we can design more powerful drugs in order to inhibit it better. And that's exactly what that paper describes. And finally, I think uniqueness is something which you find as an aesthetic principle in general. There's the uniqueness of a single rose petal. the uniqueness of anything isolated that grabs our attention in the wider world. But I think in nature and data, in biophysics, there's a sort of uniqueness even to things which show no apparent order that you can easily perceive, no symmetry, but the unique complexity of something like the ribosome and the ways in which the bits of RNA and protein come together to form that structure are, it seems to me, another way in which aesthetics is experienced by people studying nature with data, as it were. And so I've had these six kind of key ideas: idealism, observation, representation, form, fit, and uniqueness. They are all different ways, practical ways in which I think aesthetics drives or is an experience engaged with by scientists in studying nature with data and doing biophysics. And if I had to group them all together in terms of what is going on, I think that the scientists are recognizing something when they see beauty in their data or find beauty in their experimental, what the experiment is showing them or the structure they determine. And also there is a delight that they have, and this would apply to the experimental process as well as to the actual results that you achieve, and which delight you with their form, with their fit, with their uniqueness, the way they represent a new understanding to you, and with the idealism that they might elicit from you as to how this system might work. And now I'll finish.